In fact, there are some differences between the female and male skeleton. You already know some from anatomy. The female structure is structured in a way that it can actually allow the passage of a fetus throughout the vaginal canal. So that makes sense in the way that the female skeleton has a more broadened structure, a more widened structure of the pelvis, along with the bones as well of the pelvis. But in the male is more narrow, and there is different centers of gravity in both. That's just some anatomy before we start actually talking about the bone from a histological point of view. So let's start talking about the functions of the bone, generally speaking. First of all, the bone is necessary to structure and support the human body. Without a properly functional bone, there is no possibility of movement. This is because the tendons of the muscles are actually attached to the bones, and without any proper structure and proper support of the bone, there is no possibility of movement whatsoever. Of course, the human skeleton protects many vital structures, such as the brain, such as the heart, and many others. So, of course, one of the major functions is protection of the human body. The bone, in fact, houses the hematopoietic centers, which is the bone marrow. Within the bone marrow, there are the many different blood stem cells, and of course hematopoietic stem cells, which allow and which practically are the centers of where all the cells and the products of the human blood are formed. And the word hematopoiesis comes from the word hema, which means blood, and poiesis, which means to produce, to make. So it's the hematopoietic centers in the bone marrow. One of the most important functions of the human bone is that it stores minerals. In fact, it stores phosphate, calcium, and just because these two ions, especially calcium, plays a major role in the endocrine, in the whole endocrine, calcium metabolism and hormonal function, because calcium is not just any ion. It's an ion that also controls cell movements and cell functions. Calcium, in fact, is one of the ions that induces some cell actions and some cell reactions, and also it plays a major role within the balance and the concentration of calcium within the blood, which must be tightly and very specifically regulated in order to allow for these cell functions to practically be within a balance. So anything that has any structure, any organ, anything that has to do with the regulation of structure of an ion or anything within the bloodstream and within the functions and the results of these actions within the cells, this is what we call endocrine. So in the beginning, we're going to talk about a bit about the cells of the bone, what cells we have within the bone structure, and then we're going to start talking about them specifically one by one. First, we're going to talk about just shortly about the functions of each, and then we're going to go thoroughly and detailing throughout all of them one by one. So we have the osteoblasts, and as typically all of the cells that have the term blast are producing cells, are highly active cells, and osteoblasts are the cells that produce osteoid. We'll discuss what osteoid is. But if someone says osteoblasts produce bone, that's an incorrect statement. They produce osteoid. We'll discuss what it is in a bit. Then we have the osteocytes, and as always, the term cyte, especially when we have cells that the progenitor cells are blasts, are the not inactive, the less active form of this cell. In this case, the osteocytes are cells that practically maintain the structure that was being produced, the bone that's being produced already. Osteoblasts are responsible for the production of osteoid, which in time will mature into bone, and osteocytes are lodged within the bone and practically maintain this structure by producing specific substances. We'll discuss which substances they are. And lastly, we have the osteoclasts. The osteoclasts are the cells that destroy bone, that destroy the structure of hydroxyapatite and destroy the structure that we will discuss within a second. So just by seeing these three cells, we easily can identify a balance of production of bone and balance of destruction of bone. The osteoblasts and the osteocytes are responsible for the production and maintenance of the bone, and the osteoclasts are responsible for the destroyal of the bone. So let's see, like in a very small schematic, how this looks like. So the osteoblasts are cuboidal or columnar cells, depending on the amount of production. So these cells are typically within the periphery of the islands of osteoid. We'll discuss what this is. and they typically have this cuboidal-columnar active appearance, meaning that they have excessive endoplasmic reticulum, rough endoplasmic reticulum, and Golgi apparatus. And of course, we have within the osteoid, you find the osteocytes, which are the ones that maintain the structure by producing ECM and other molecules. We'll discuss them in a bit. And lastly, we have the osteoclasts. The osteoclasts, as I was saying before, destroy the bone itself. So this is pretty much a schematic of how this all looks like within and like animated. And this is the true histological picture. We'll go through them in detail in a bit. So let's start talking about a bit the osteoblasts. How do these cells produce, what they produce, and how these cells produce osteoid? So osteoid is nothing more than a collection of ECM that we discussed in the cartilage as well, and the connective tissue. In this case, we have type 1 collagen, as always. This is the fiber of the bone, very important structure. Then we have the glucosaminoglycans, proteoglycans, and multi-adhesive glycoproteins. The same 3-4 features that always make up the ECM. In the beginning, we have the fibers and the amorphous ground substance. The amorphous ground substance is the glucosaminoglycans, proteoglycans, and glycoproteins. So the osteoid is nothing more than an aggregate of these structures of type 1 collagen and, of course, many other enzymes that start to aggregate hard ions, which is calcium and phosphate. So within the most important thing is this interaction between collagen and the osteoid and the enzymes of the osteoid. So collagen practically plays a role. Type 1 collagen is nothing more than just lines of collagen, lines of fibers that in the end will become aligned. Why do we care about this? Why do we care about this alignment? Well, because it's not just the density of these ions and the hardness of these ions of calcium and phosphate that make the bone also extremely resistant structure. In fact, it can actually withstand within 1 ton of weight, 1 ton of kilos. This is insane. So it's not just enough to have only these very hard ions. We'll discuss the ion. The ion that they form is hydroxyapatite, the same of the teeth and many of the hard tissues of the bone, hard tissues of the human body. I'm sorry. So we have first of all the alignment of the collagen as it is. Let me show you this picture that actually helps a lot of how this works. So we have the osteoblasts that produce these 3, let's say, main substances: type 1 collagen and the 2 enzymes that we're going to talk about right now. So we can see that the ions, that these lines represent the collagen fibers. So we have production of a lot of collagen type 1. So in the beginning, this is just irregular and irregularly structured, not parallelly, not anything, no specific organization. So first we have the deposition of collagen. Then within this, let's say this matrix, this osteoid, this is the osteoid in fact. So within this osteoid, we're going to have the collagen that's being started to be attached by 2 different enzymes. We're going to have the osteocalcin. This is the enzyme, osteocalcin. Osteocalcin, as the name suggests, is the enzyme that actually attracts calcium ions. And then we're going to find the matrix vesicles. These circles are the matrix vesicles. The function of these vesicles is that they simply contain this very, very important enzyme, one of the many that they contain, but the most important enzyme they contain is alkaline phosphatase. And again, as the name suggests, alkaline phosphatase, this is the enzyme that actually attracts phosphates, the phosphate groups. So we have the osteocalcin at one point that attracts calcium, and on the other hand, we have the matrix vesicles that contain the alkaline phosphatase, which practically aggregate phosphate. Whenever you have this aggregation of calcium and phosphate groups, they typically tend to form this very, very strong structure. This is the molecule of hydroxyapatite. So the issue is the point of this function, the point of the osteoblast is to produce the fibers on which the calcium and the phosphate will come, aggregate, and form the hydroxyapatite. So after we have this attachment of these ions on the collagen type 1, then this is going to start to practically aggregate these ions, form this crystal, this very rough crystal, and start to become parallelly arranged. This parallel arrangement of type collagen 1 along with the ions, along with the crystals of hydroxyapatite, this is the structure that actually makes our bone so resistant and so resilient to stand and withstand tremendous amount of forces. So in summation, Osteoblasts are the cells that produce osteoid. Of course, within the osteo we find type 1 collagen and matrix vesicles along with osteocalcin. These three components come together to form parallelly arranged collagen fibers reinforced with crystals of hydroxyapatite. Everything clear so far? So this with this structure right here, this is the parallelly arranged hardened osteoid. The hardened osteoid practically makes the bone as it is. So before this happens, we have this this point right here, this small vessel, this small granules that you see in this picture. This is the osteoid. This tiny structure is the osteoid. Whenever we have osteoblasts entrapped within the osteoid, this is a very common characteristic, that after deposition of this osteoid, then we're going to have many of these osteoblasts found within this structure of osteoid. They will become entrapped by hardened structure, by this hardened bone structure. So this is going to, anytime an osteoblast is surrounded by hard material, which is exactly what we discuss about right now, with osteoid that eventually will mature and harden, this cell, this osteoblast that is encircled, is going to become the osteocyte. So again, the osteocytes, the progenitor cells of the osteocytes are the osteoblasts. So we said shortly the function of the osteocyte, which is to maintain the structure of bone. What does that mean practically? Practically means that this osteocyte, this former osteoblast, now osteocyte, is going to still keep on producing many substances of the ECM. It means, again, type 1 collagen, and again, the glucosaminoglycans, multi-acidic glycoproteins, and protoglycans. What is the purpose of this cell? The purpose of this cell, that it's because it has a strategic placement within the bone, it practically is able to connect many of the different osteocytes together, because it has extensive cellular processes called canaliculi, and these cells reside within the lacuna. So these spaces are called lacuna, and the cells that reside within this lacuna are called the osteocytes. The function of this osteocyte, as I'm discussing right now, is that it maintains the homeostasis. What does it mean, homeostasis? Again, homeostasis is a balance. It's a stable balance. This has to do with the calcium. This has to do with how much calcium is deposited within the bone and how much calcium is going out of the bone. In a very simplistic terms, the osteoblasts and the osteocytes are responsible for how much calcium is within our bones. The more calcium you have within your bones, the less you have in the bloodstream, and vice versa. The more calcium you have in your bloodstream, the less calcium you have in your blood. The more calcium you have within your bloodstream, the less you have within the bone itself. So the bone is the ideal storage of calcium, and of course phosphate as a second role, but primarily calcium. So these two cells, osteoblasts and osteocytes, are responsible for this amount within the bone. So these cells, because they have this strategic location, are able to play a role, of course, in the calcium homeostasis, but also they're responsible for the production and the rate of destroying of the rate of collapsing and destroying of the bone, and the rate of producing. Meaning, how do they do this? Well, because at any moment, any given moment, whenever you have the activation of a cell, either mechanically or chemically, this cell is still active, meaning that these cells work as mechanical receptors. That's why you have many people in old age that they're not really mobile in their old age. That's why many doctors actually suggest that people of old age work out more, take more walks, do more exercise. This is because they want to prevent the osteoporosis. Osteoporosis is the disease that the bone shrinks, and the thickness and the hardness of the bone is being minimized. And this is because people that do not work out, people that do not have an active physical life, meaning they have less mechanical pressure applied on the bone. And the less mechanical pressure you apply on the bone, the less stimulated the osteoblasts and the osteocytes are. The less stimulated the osteocytes and osteoblasts are, the less deposition of osteoid, and again hard tissue is produced within the bone. So these cells play one of these very important roles of mechanical sensors. So they sense a mechanical stimulus and induce and activate furthermore their productive phase. If someone has a keen eye, you would notice that in both of these sections of the slides of osteoblasts and osteocytes, I talk about cytokines, that these two cells produce cytokines. Cytokines are typically molecules that activate and induce other cells, either formation or action or even reaction. A typical example is a cytokine production from the inflammatory cells. For example, meaning that the monocytes, the macrophages, neutrophils, and of course all of the different types of cells produce cytokines. Some different forms than others. Some cytokines have different functions than the others. But a typical example is that of inflammation. Whenever you have a pathogen within anywhere in your body, your immune cells produce cytokines and induce the inflammatory changes. So again, cytokines is a very big group of molecules that activate and induce either cellular aggregation, cellular collection, or cell action or reaction. Keep this in mind. This is going to be very, very useful. That the osteoblast, the fact that the osteoblasts and osteocytes produce cytokines is of major importance. We'll see in a second how actually this is really that important. So far we discussed about all these two cells that produce the osteoid and eventually are responsible for the production of bone. Let's start seeing the other edge of how this bone is destroyed and again produced. Let's see how this bone is destroyed. We have these osteoclasts. The osteoclasts are in fact very, very big cells that have typically 5 to even 50 nuclei. This is not typical for cells. The only other cells that you'll see in the human body that have more in normal conditions, the only other cell that has more than one nuclei is the skeletal muscle. We'll discuss this in our muscle lecture. But so far, this is the first cell that we meet that has more than one nuclei. So the function of the osteoclast is to destroy bone. How does this work? How does the osteoclast, just a cell, destroy one of the most difficult and one of the most resilient structures in our human body? Well, these cells practically are again within the bone, on the surface of the bone. We'll see where they are located, and they form these two structures that are very, very important on destroying the bone. First, they form this hair-shaped lacunae, or also called resorption areas. This area that practically is the location where the osteoclast attaches and grabs on the bone. So in the beginning we have this formation of the osteoclast, and the second stage is that these cells practically attach on the bone surface. After they attach on this bone, then they will start to form this the cell processes of the osteoclast will start to form these wave-like projections. And again, we've seen in previous lectures that this wave-like structure serves one purpose: to increase surface area. So we have the structure that the osteoclasts project these cell processes on the bone, and as a consequence they have increased surface area that attaches on the bone. So the thing is, okay, they attached, they're stable, and within the hair-shaped lacunae, they're on the bone. What next? How do they destroy the cell? How do they destroy the bone? So as we know, calcium and phosphate are both ions that are of bases. Both calcium and phosphate, phosphate not so much, but calcium typically creates a very basic environment. So what this cell does is that it practically shoots and throws high concentrates of protons, of acids, of very potent acids on this surface of the bone. So this acidity is the thing that in the end will neutralize, will destroy the structure of hydroxyapatite. And after this hydroxyapatite is destroyed, then we have the collapsing of the structure of collagen. So the first stage to destruction of bone is practically destroying and tearing apart the hydroxyapatite. And this is done through very potent acids. Here you'll see the term carbonic anhydrase. Keep this enzyme in mind. This enzyme actually is very useful in more than one organs and more than one cells. Typically, you'll see this cell also in the erythrocytes. The erythrocytes, of course, the function of erythrocytes is to what? To bring oxygen and to practically get rid of carbon dioxide. The same enzyme can play many different roles and many different functions. So in the erythroblast, this carbonic anhydrase plays a role in the carbon dioxide reading. In this case, it plays a role within the formation of acid. So of course, the calcium hydroxide, the osteoblast uses this enzyme to produce potent acids to destroy the bone. Question is, okay, we discussed so far how it destroys it, how it binds, but how it binds the bone, it attaches on the bone, and how it actually destroys the bone. The question is, how is it formed, and how does this cell have more than one nuclei? Typically, the cells that have more than one nuclei are fused cells. So in this case, the osteoclasts are fused cells. Does anyone have any idea what kind of fused cells they are? Let me help you a bit. Before we discussed about the cytokines. The osteoclasts are practically fused monocytes. We discussed about the monocytes in the connective tissue lecture. So, how do these monocytes know where to go? How do the monocytes that are typically reside in the bloodstream, they know where to go and how to actually, why fuse? This is the job of the osteoblasts and the osteocytes. So practically the highest amount of cytokines is produced by the osteoblasts. Meaning what? Meaning that the osteoblasts produce cytokines. These cytokines as a consequence, they induce the aggregation of the monocytes, and these monocytes fuse together to form the osteoclast. What does this tell us as a consequence? It tells us that both the rate of production of bone and the rate of destruction of bone is tightly controlled by osteoblasts. Practically, the cells that produce bone are the ones that actually play the major role and control the rate of destruction. This is why these cells have 5 to 50 nuclei, because they are practically fused monocytes, aggregated and controlled, orchestrated by the osteoblasts. So here we see a nice picture of how this actually looks like macroscopically. We'll see, of course, all the pictures after we finish the theoretical part of this lecture. So this is the osteoclast. We can typically see these 3 nuclei. All of this is one cell. All of these nuclei reside within the one cell, so it's very easy to identify an osteoclast in the macroscope, simply because we just see more than one nuclei on the surface of the bone. So this is very easy to identify, and this is the electron microscopy picture of how this structure looks